Myosin II ATPase is a hydrolase enzyme that takes part in muscle contraction. We will be presenting an article that is studying the mechanism by which this occurs and presents a novel hypothesis on the mechanism.
Myosin II ATPase is a kinase protein that belongs to the superfamily myosin. This protein binds actin filaments and pushes it or rows it. This movement of actin filaments causes muscle contraction that are used for movement. When myosin binds to the actin, there is a conformational change in the myosin head, causing it to alternate between 90 degrees relative to the neck and a 45-degree orientation. In other words, the head binds the actin filament at its 90-degree orientation. Then ATP, which is bound to the myosin head, is hydrolyzed to ADP, which causes the bound head to switch to its 45-degree orientation. This change pushes the filament before the head unbinds, and a new ATP molecule binds. The myosin and the head return to its 90-degree orientation, where the cycle begins again. This cycle is called the Limb-Taylor cycle.
Myosin II is a part of an enzyme superfamily of proteins. It functions to hydrolase ATP, bind to actin, and force transduction. The protein is composed of two heavy chains and multiple light chains, organized into three structurally and functionally different domains: the head, the neck, and the tail.
The head is composed of heavy chains. The head functions as the location of actin and ATP binding and generates the force required for conformational change that propels the myosin along an actin filament, and thereby acting as the ATPase. This domain corresponds to the end terminus of a heavy chain. The neck is composed of light chains, both regulatory and essential, and regulates the activity of the head domain. The third domain, the tail, determines the role of each protein in the myosin family. In myosin II, the tail is composed of heavy chains that form a coiled coil structure, resulting in a relatively long, rod-like structure. Multiple molecules of myosin II will pack together by means of their tail domains to form a thick filament. Here the tail domains form one large rod with multiple head domains protruding, able to react with actin filaments. This domain corresponds to the C-terminus of the heavy chain.
Is a member of the myosin kinase superfamily. Within this superfamily, the enzyme in the myosin family. Myosin is a hydrolase. Protease cleavage has been used to determine the full domain structure of myosin. Typically, proteases cleave myosin at the base of the neck domain, producing a rod-like tail fragment called the light meromyosin, and at the head and neck fragments called the heavy meromyosin. Further digestion is required to separate the head and neck domains. Reacting the heavy meromyosin with papain separates the neck fragment from the head domain and isolates the two heads of the molecule.
Myosin II is a hexameric protein. This refers to the number of subunits or complete peptide chains that are linked together to form the protein. A simple description of the structure of this protein has the head domain at one end, with the end terminus ends of the heavy chain intertwined with the four light chains that lead into the neck of the protein. Extending from this bundle of chains is the coiled coil structure of the two heavy chains that form the tail domain. The end of the tail has a shorter end at a different angle. This is the part that binds to actin and will transition between a 90-degree and 45-degree angle from the rest of the tail domain.
The Limb-Taylor cycle is the mechanism describing the function of the myosin. Actin acts as a stimulant and nucleotide exchange factor, stimulating the hydrolyzation of the ATP, providing the conformational change of the molecule. Myosin forms a cross bridge that is bound to the actin in the rigor or 45-degree down position. At this stage, the active site is considered open. ATP binds the ATPase site on the myosin head, quickly dissociating myosin from the actin and changing from the open to the closed position. Hydrolysis of the ATP produces ADP, inorganic phosphate, leading to the return of the myosin to the myosin cross bridge to the 90-degree up position, where it rebinds the actin to a new location at the filament. This causes the release of ADP and returns the myosin head to the 45-degree state, rowing the actin along as it is still bound to the myosin head. It has been hypothesized that only a portion of this actually moves, providing an explanation for why visualizing this conformational change has historically been so difficult.
Myosin II ATPase, commonly denoted as a gene as MYH2 in humans, contains 1,942 amino acid base pairs that are localized on chromosome 17 in humans. These amino acids make up 6 peptide chains that join to form the protein. Myosin II has a similar amino acid sequence to multiple proteins in the myosin family. It is very closely related to myosin I in the number of and species of amino acids in its polypeptide chain. Myosin II contains 1,941 amino acids, and myosin I contains 1,939. Between the two, 95% of the identities of the amino acids match up in the chains. Myosin V, on the other hand, contains only 1,219 amino acids and only identifies with 35% of the amino acid residues within myosin II.
There's one slight variant of our enzyme known as non-muscular myosin II-C. This molecule does not require the regulatory light chain for the maximum efficiency of actin binding.
Structurally related proteins, such as those others in the myosin family, perform a similar function to myosin II. Myosins interact with actin filaments as motor proteins. Muscle myosins contract muscles through this interaction, binding to the actin through their head domain. Different myosin genes are composed of differing amounts of heavy and light chains, but form the same basic domains of head, neck, and tail. For example, myosin I has a very similar mRNA structure to myosin II and has a head and neck domain, but is lacking in a tail domain. This protein also participates in muscle contraction, actin filament binding, and is therefore considered a motor protein. Myosin V has a similar head and neck domain.
Are studying the mechanism by which myosin proteins in general, as a whole, bind, move, and manipulate the myosin-actin filament. Understanding myosin II specifically would then allow us to understand the broader mechanism by which myosin proteins as a family carry this reaction.
Actin filaments are linear polymers of globular actin, found as microfilaments in the cytoskeleton and thin filaments in muscle and non-muscle cells. Within muscle cells, they are aligned for myosin proteins to push the filaments, known as thin filaments, along, causing a muscle contraction.
Myosin II has 3 major parts. The S1, or the head domain, which is the binding site for the actin filaments, made of heavy chain. The neck domain, made of light chains, and the tail, composed of heavy chains. There are 2 heavy chains in each myosin II molecule that each comprise a head connected to one coil on the tail. There are 2 kinds of light chains in myosin II, and 2 of each located at the base of the myosin head. Regulatory and essential chains function in calcium 2+ binding, stiffening the neck and preventing a conformational change in the myosin head. However, the exact method of this function is unknown. It has been determined that serine 19 on the light chain is phosphorylated, causing the head to contract and the neck to stiffen, preventing binding. Phosphate kinase regulates this process by dephosphorylating the regulatory subunit. Essential chains are even less well understood, though it is predicted that they participate in calcium 2+ binding as well to stabilize the myosin head.
It is suspected that myosin bind cooperatively, meaning that bound myosin heads heighten the affinity of nearby heads for the actin filament. In low ATP environments, this tendency causes the heads to bind in clumps on the actin, leaving other parts bare. This reflects the role of favorable conformation of actin filaments on the relative affinity of binding myosin heads. However, this variability is difficult to detect in vivo due to low affinity in the presence of ATP. This study utilized GFP as a stain to visualize infused myosin heads with amino acid substitutions that would heighten the affinity for actin filament binding in the presence of ATP. The goal being to identify subsets of actin that myosin II would preferentially bind to. They were successful in discovering myosin heads preferred mechanically stretched sections of the actin filament while in vivo conditions existed.
The soil-dwelling amoeba Dictostelium discoideum was used for this study. The electrocompetent cells prepared from this species were transformed with the stain and the HL5 gene, which is a light chain 5. Utilizing confocal fluorescent microscopy, live cell observations were made of the GFP mutant head and the GFP mutant myosin II heavy chains. Cells were allowed to settle to the bottom of the petri dish and scanned with a confocal laser scanning unit. This method of microscopy scans point by point, allowing multiple depth viewing and essentially a three-dimensional rendering of the sample. Cells were starved for 8 to 10 hours to induce chemotaxis. This is important for stimulation of the amoeba to move towards something, allowing the viewing of the actin stretching. The same method was used to observe fixed cells on glass bottom dishes, made permeable and fixed by replacing calcium sodium phosphate buffer with a solution of pipes, magnesium chloride EGTA, DTT, Triton X100, and glutaraldehyde. After fixing the cells for 10 minutes, they were stained for 1 hour in PBS. Rinsed in PBS containing DTT, and then finally observed through the confocal laser scanning microscope.
Two types of stains or probes were needed to detect actin filaments with higher affinity for myosin II motor domain. One that would accurately report local concentration of actin filaments, and one that would preferentially bind to subsets of actin filaments having higher affinity for myosin II motor domain. In the report of local concentration of actin filaments, the stains compared were GFP actin binding domain, ABD, and of actin binding proteins, and RHPH. GFP actin and RHPH were found to be very similar, while GFP ABD and RHPH were dissimilar. RHPH was chosen as the actin stain for the study as it allows the total actin filaments to be quantified.
As for the preferential binding of subsets of actin filaments with higher affinity for the myosin II motor domain, the use of fluorescently labeled motor domain lacking a tail domain was required. This was done because actin filament formation involving the tail domain may localize myosin II filaments in vivo. At physiological levels of ATP, the head only weakly associates with actin filaments, a mechanism demonstrated by the Lemn-Taylor cycle, a slow actin-stimulated release of ADP-PI from the head establishes a strong binding in the actin filament, followed by a rapid powered stroke, or the rowing action, that is almost immediately followed by the dissociation of the actin filament from the head. This results in a small amount of time that the head and the actin filament are actually associated. For this reason, myosin II ATPase, designated G680A, was incorporated into the head protein and fused N-terminally to GFP. This mutant is one of many that is made with an extended, strongly bound state in the presence of ATP.
These trials found that both myosin head mutants showed unique localizations along the actomyosin cortex, particularly around the front and rear of the cortex. From this, it was inferred that the GFP-S1 mutant stained myosin II head could provide consistent results, and was therefore chosen for the probe required to show affinity higher than naturally occurring myosin II head. The patterns of the GFP-S1 mutant cells were permeabilized and fixed in the presence of Triton X100 and glutaraldehyde, then stained with RHPH and GFP, and the resulting fluorescence appeared similar in all cells examined. This comparison of patterns between the two stains showed reproducible differences in the distribution of bound S1. Figure 4 displays the differently stained samples and their signal intensities. A shows multinucleated P10 phosphate and tensine homolog undergoing cytokinesis. The far left image is a GFP stained fluorescence image, the middle RHPH stained fluorescence image, and the right image displays the two images superimposed upon each other. B and C display mutated cells undergoing cytokinesis. Preferential binding of certain regions of the actin filament to the myosin II is visible in this section when compared to A. Instead of the evenly dispersed coloration seen in A, these cells show spots of coloration, and regions along the actin filament left gray and uncolored.
This paper introduced 3 proposed mechanisms for preferential binding of myosin II to the actin filament. Myosin transiently forms bipolar filaments and associates with specific subsets of actin filament to drive local contraction of the cellular cortex. Myosin II filaments must selectively bind these subsets of actin. The first proposed mechanism begins with a mechanosensor triggering a biochemical pathway, leading to enhanced affinity of actin filaments for myosin II. Isoforms of tropomyosin have been shown to differently regulate affinities of different actin filaments for myosin heads. However, the genes for this protein do not exist in the genome for the organism used in this study, making this speculation irrelevant for the findings of this experiment. The next proposed mechanism assumes that during cytokinesis, a stretch is induced upon the actin cytoskeleton, aligning the filament for binding with myosin II. In the absence of ATP, myosin tends to form bundles of actin filaments. Therefore, it is possible that a GFP-S1 carrying a mutation heightening its affinity for actin in the presence of ATP prefers binding to be aligned or stretched areas of the actin filament. Upon the cell receiving a mechanical stimulus and beginning to move, the filaments are stretched, causing a conformational change in some of the actin subunits.